at these cute baby animals, you will have immediately observed how cute and fluffy they are. But you will also have noticed that they are different. They vary, yes, some are puppies and some are kittens. There is a great deal of variation between species, but also there is variation within a species. One puppy is fluffier than the other. They have different color fur, and the kittens have different color eye. Variation is the difference in the characteristic of individuals in population. It can be due to genetics and the environment, or a combination of both. Let's start with genetics. Different members of a population have different DNA, unless they are identical twins. This means they have different versions of genes called alleles. And these alleles give rise to different characteristics. For example, on the eye color gene, this kitten has an allele for blue eye color, whereas this kitten has an allele for brown eye color. On this note, let's understand chromosomal mutation. As the name says, chromosomal mutation means the mutation of chromosomes. And how exactly does it get mutated? It can happen in two ways. One way is where the structure of the chromosomes gets disrupted. The other is where the number of chromosomes are different than the usual in the organism. Let's have a look at each type. This is the structure of a typical chromosome. Now imagine this small part gets physically removed. This will result in the chromosomes getting smaller as its segment is lost. This also means if the segment had some important genes, then they will also get lost, which can result in severe abnormalities. Such type of a mutation is called deletion. This is where the part of the chromosome gets deleted. Now imagine this part of the chromosomes get duplicated. As a result, the chromosome's length increases. This will also result in some or the other abnormality in the individual. This type of mutation is called duplication. Now comes the next type. These are two different gene segments. Imagine they get flipped. Thus, the chromosome will appear like this. This is called inversion. Do you think this type of mutation is harmful? It may or may not be harmful depending on which genes get flipped. Now let's take one more chromosomes. Imagine a part of this chromosome gets removed and gets inserted in this chromosome. What will be the result? That's correct. Both the chromosomes will change drastically. This type of mutation is called insertion. And now comes the last type. Instead of a segment getting inserted to another chromosome, let's imagine a situation where a segment from each chromosome gets interexchanged. So this segment will get into this chromosome and vice versa. This kind of mutation also results in abnormalities as both the chromosomes get altered completely. Any idea what this type will be named as? Well, this type of mutation is called translocation. All this mutation resulting in the disruption of normal chromosome structures abruptly happened under a single heading. They are categorized as chromosomal aberrations. Now this was about the mutations that involve change in the structure of the chromosomes. Deficiencies or deletion. A chromosomal deficiency occurs when a chromosome breaks and a fragment is lost. Chromosomal deletion can be detected by a variety of experimental techniques, such as cytological molecular probes and genetic analysis. Genetic deletions can be revealed by a phenomenon known as pseudodominance. One copy of a gene is deleted, so the recessive allele on the other chromosome is now expressed.
The chromosomal will duplicate part whereas the abnormal genetic events that cause a gene duplication over the course of many generations. That two genes may differ due to the gradual accumulation of DNA mutation will cause duplication. The one copy of the one normal of chromosome and one copy of an inverted chromosome will call it inversion. An insertion is a type of chromosomal mutation where a segment of DNA is added into a chromosome. This extra DNA can come from the same chromosome, a different chromosome, or be duplicated from existing genes, we call it insertion. A chromosomal translocation occurs when a segment of one chromosome becomes attached to another. In reciprocal translocation, two non-homologous chromosomes exchange genetic material will come balanced translocation. In simple translocation, the transfer of genetic material occurs in only one direction. Will be imbalanced translocation. So we call it translocation. Next is change in the number of chromosomes. And euploidy will presence of an abnormal number of chromosomes in a cell. Trisomy 21. In this condition, the majority of chromosome 21 is attached to chromosome 14. The individual would have 3 copies of gene found on a large segment of chromosome 21. Therefore, they exhibit the characteristic of Down syndrome.
Human beings are made of millions of cells. Inside every cell lies a nucleus which has 46 chromosomes. A chromosome carries genes which make a center human being. A chromosome are numbered from chromosome 1 until 22, which chromosome 1 being the largest chromosome, and chromosome 22 being the smallest, and the last parent's chromosome are sex chromosomes, with boy having X and Y, and girl having 2 X chromosomes. Homologous segregation refers to the separation of homologous chromosomes during meiosis 1, specifically in anaphase 1. In a diploid cell, chromosomes exist in pairs called homologous chromosomes, one from each parent. Non-homologous chromosomes carry different genes. For example, chromosome 2 may carry genes for blood type, while chromosome 7 may carry genes for cystic fibrosis. Errors involving non-homologous chromosomes like translocations can cause genetic diseases or cancer. A chromosomal translocation is a genetic change in which a piece of one chromosome breaks off and attaches to another chromosome. There are 2 types of translocation: Robertsonian translocation and reciprocal translocations. The most common is a Robertsonian translocation, which is found in about 1 in 1,000 people. In this type of translocation, the long arms of 2 acrocentric chromosomes fuse together, causing us to lose the 2 short arms. As we learned in the previous section, 13, 14, 15, 21, and 22 are all of the acrocentric chromosomes in our cells. Let's go through an example together. The most common Robertsonian translocation takes place between chromosome 13 and 14 and makes up approximately 75% of all Robertsonians. As mentioned in the previous sections, an affected person has 46 chromosomes. As you can see here, if you are a carrier of a 13, 14 or Robertsonian translocation, you will have 45 chromosomes instead of 46. You will have 1 full copy of each of chromosome 13 and 14. You will also have a fused chromosome, which is the long term of 13 fused to the long arm of 14. When looking at the difference, we can see the only big difference in a Robertsonian translocation is the location of the long arms and the 2 missing small arms. How does this happen? This could be either be passed down from one of your parents, meaning that they are also a carrier of Robertsonian translocation,